Hey everybody, so we're going to take a look at our review sheet for genetics. Now in this review sheet, the different types of Punnett squares genetic crosses that you guys are going to be working through on your final exam, they appear on this study guide. So we're going to work through these together. Hopefully you've worked through this on your own first. I recommend that. So if you have not touched this sheet at all, I recommend seeing what you can do on your own first. So stop this video right here and hop over to that sheet and work through what you can, and then come back and watch the video and check your answers, okay? Check your answers, and if there's something that you miss or don't understand, hopefully this review will catch you up, okay? So we're going to jump in right here at the very first part, monohybrid crosses, our part A, vocabulary. So dominant, recessive, homozygous, heterozygous, what does all this mean? So first up, I'm going to zoom in a little bit to make this a bit bigger. Hopefully I won't give you motion sickness. Sorry, it may not be even, maybe crooked. Okay, so circle the dominant alleles. So first off, what is an allele? An allele is a representation of a trait, okay? We use letters to represent the alleles. So you see these letters, D, A, E, T, B, P, S. I'm on number 1 right there. So these letters are alleles, and they represent traits, genetic traits. Dominant and recessive. Dominant means the trait will be expressed. Recessive means the trait can be hidden. Okay, so since dominant, we think of something that's dominant, puffed up, we represent dominant traits by capital letters. It's just symbolism. So a capital letter, if you see a capital letter for an allele, it's a dominant trait. If you see a lowercase letter, then that means it's a recessive trait. So very easy, straightforward question right here. Circle the dominant alleles. Hopefully, if you've worked through this, you're circling all the capital letters, right? D, T, P, S. We don't know what traits these letters represent, but if they're capital, we know they're going to be dominant. Number 2, circle the recessives. So all of this is going to be your lowercase letters. So lowercase a, T, W, and R. Those are our recessive alleles. Next up, homozygous dominant genotypes. Now, let's break down the vocabulary word in this question. Homozygous. Homo means the same. That's a medical term. Whenever you see the prefix homo, that means the same. So homozygous means same alleles, okay? Then next up, that's homozygous. I'm going to underline that word right there. Same alleles. Then our next adjective is dominant. Well, we already know dominant's going to be our capital letters. These are traits that will be expressed. So they're capital letters. Genotype is simply your alleles. That's your genes, your alleles. So circle the homozygous dominant genotypes. We're just looking for 2 capital letters, friends. So capital G's here, capital T's here, capital A here. That's our answer. Just 2 capitals, okay? Number 4, circle the homozygous recessive genotypes. There's our word homozygous again, which means the same. Recessive now means that the trait will be hidden or overshadowed by a dominant trait. So remember, recessive is our lowercase letters. That's going to be our lowercase letters. So we're looking for 2 lowercase. Here's 2 little g's. Here's 2 little r's. And here's 2 lowercase d's. There's our homozygous recessive genotypes. Last question, number 5, circle the heterozygous genotypes. Hetero, there's the prefix. Heter means different, not alike. So hetero means you're not going to have 2 capitals or 2 lowercases. Instead, you're going to have 1 of each. So 1 capital, 1 lowercase, 1 dominant, 1 recessive together. That's what heterozygous means. So heterozygous would be capital D, lowercase d, capital W, lowercase w. And that's it in that question, okay? So a little vocabulary there, being able to recognize the alleles based on their descriptor. So what's dominant, what's recessive, what's homozygous, what's heterozygous, okay? All right, we're going to jump down to part B. working through some of these Punnett squares. So I'm going to move this page again. Try not to give you motion sickness. Whoops. See, I'm doing this on my iPad with my fingers, so I'm spinning it by accident. Okay, maybe that's good enough where you can kind of see it pretty clearly. And we're going to get started. Actually, I'm going to move it over a little bit because I'm kind of blocking the bottom question there, part B. So there we go. Maybe that's close enough straight. All right, so let's look at these. Part B, monohybrid Punnett squares. Mono means one, so we're doing some crosses with just one trait in a monohybrid cross. That's when you just make your 4 square box. So let's look at our question. In guinea pigs, short hair, S, so it's telling us the letter S to represent short hair, is dominant to long hair. Complete the following crosses, fill in the blanks beside each Punnett square with fractions and percentages. So here's what I like to do. I always like to make a key. It just helps me keep what letter represents what trait straight. Okay, so we're going to say short hair is dominant. So I'm just going to come over here kind of to the margin, to the side, and I'm just going to say capital S equals long. Okay. No, sorry. I just messed that up, didn't I? I hope y'all noticed that. I was kind of reading the question. I was like, it says dominant to long hair. It's not long. It's dominant to long hair. So let me go back. Capital S is short. Okay, capital S is short. We probably would have just used the letter L to symbolize long, right? If it was dominant. So this is short hair. Short hair is dominant to long hair. So that means lowercase s is going to be long. Now here's a question that sometimes pops up. Why don't we use an S for short and an L for long? Well, because different letters represents different traits. We're not talking about different traits here. We're talking about the same trait, hair. That's the trait we're talking about. And the letters of this one trait represent the characteristics. So short versus long. It's all hair in the guinea pigs, okay? So if you were to have an S for short and an L for long, looking at that means the S represents one trait and the L represents another trait, and that's not correct. The same letter represents the same trait. So capital S dominant to lowercase s. Short dominant to long hair. So use the same letter per trait. This gets confusing when you get into dihybrid, trihybrid, quad hybrid crosses. Here's the good news. You only have to go up to dihybrid. But there is a such thing as trihybrids and quad hybrids, and you can go up and do some really complex Punnett squares, huge Punnett squares. And if you're using different letters to represent characteristics, it's going to be a hot mess. So you've got to use the same letter, okay, per trait. So this question is a monohybrid, one trait that we're talking about. We're going to use one letter. So capital S short, lowercase s long, complete this cross. It gives it to us right here. So we've got a heterozygous times a recessive homozygous. So it really doesn't matter what you do left versus across the top. I like to just start with the first individual here, the first genotype, and go down the left. So I'm going to put a capital S here and a lowercase s here. That's this one. Okay, and then I'm going to break these alleles apart right here, lowercase s, and then all you do is fill in your squares. So capital S, lowercase s, capital, lower, and then I've got 2 lowercase s's in these 2 squares. So all I'm doing is bringing this over and this down, and then over and then down, okay? Same scenario here, and filling in these boxes. So we've got a little blank over here. Short hair. Our question says, fill in the blanks beside each Punnett square with fractions and percentages. So read our instructions, fractions and percentages. So I've got 4 total. So how many out of the 4 do I have with short hair? Remember, short hair is dominant over here. So anything with a capital S is going to be short hair. So I've got 2 out of the 4, right? 2 out of the 4. I'm going to, I I was going to circle it. I don't know if I can change the color right here. I'm looking at this one and this one that have the capital letters. So that's where this 2's coming from right here. 2 out of the 4. You could break that down and say 1/2 if you wanted to simplify, right? 2/4 or 1/2, okay? And then it also asks us to do this in percentages. So 2 out of 4 or 1/2 is simply 50%. So 50% of the offspring, remember we're filling in the boxes, these are the offspring. So 50% of the offspring are gonna have short hair, which means the other 2/4, 50% are gonna have long hair. So now if we look at the little s's here and here, we're gonna end up with 50% or 2/4, the other half, long hair, 50%, okay? So that's our first question. Part B, both parents are now heterozygous, all right? Now, it doesn't give us the parents in this one, so we have to know what heterozygous means. You know this, hetero means different. So if both parents are heterozygous, they're gonna have different genotypes. So it's gonna be a capital and a lowercase. Homozygous would be the same. Heterozygous would be different, a big S and a little s. So I'm gonna come down the side with one parent. If you wanted to come up here and just for, you know, your brain to kind of keep things straight, write the parents right here. That's what heterozygous is. So now I'm gonna break down one parent down the side, capital S, lowercase s, the other parent across the top, capital S, lowercase s, and now I'm gonna fill in my Punnett square, big S, big S, big S, little s, big S, little s. I like to put capital first. It just helps me keep things organized and it is a visual cue when I'm trying to identify genotypes and phenotypes. It is not incorrect if you were to put the lowercase s first. It just is harder to interpret. Again, when you start to get into these big crosses, if you can have a routine and be a bit more organized in how you're documenting the alleles and documenting the offspring, it's just easier to interpret. You're less likely to make mistakes. So you noticed I put my capital S first here. And then I'm gonna have lowercase s here. Same question, fractions and percentages. So now, for short hair, you know what? It doesn't tell us. What I did here was the genotypes, and I'm going back and I'm not necessarily, oh yeah, it is asking for phenotypes, I suppose, because short and long are the phenos. So 50%, 50%. So let's go by the phenotypes here. What percent's gonna have short hair? Remember, short hair is dominant. Anything with a capital S is gonna have short hair. So I've got 1, 2, 3 out of the 4. So 3/4 are gonna have short hair. And if we were to represent this by percentages, that would be 75%. 3 out of 4, 75%. Think of quarters. 4 quarters make $1, right? 3/4, 75 cents. So 75%. And then for long hair, I'm just gonna have this little fella down here for long hair. That's the other 20, whoops. Let me do it as a fraction first. Jump to 25%, erase that. 1/4 as a fraction, and that would be 25%. All right, we're jumping to question number 2. Looks like we've got a different trait coming up. Heterozygous smooth pea pod plant is crossed with a wrinkled pea pod plant. List the genotypes of the parent. Set up Punnett square possible gametes. All right, so let's create a key. I always like to do that first so I know what letters representing what traits. We can use S again for smooth. We're going to put, it doesn't really distinguish in this question that which one's dominant and recessive, but we're going to call smooth pea pod dominant just because it is, and a wrinkled pea pod plant as recessive. So I'm going to do a capital S equals smooth, and then a lowercase s in this case means wrinkled. Okay, now don't get this confused with the previous question. We did short hair and long hair and we used the letter S. The context of the question tells you about the trait. So we used S to represent short hair because short hair was dominant, and we just picked the letter S because it's for short, okay? Same thing here. Smooth is dominant. So we're gonna pick a S just to represent the word smooth. And we have context of what the trait is in our question that's here. All right, so capital S for smooth, lowercase s for wrinkled. Those are our alleles. Now, the question says it's heterozygous. Heterozygous smooth pea pod. Heterozygous smooth. The fact, now how you might be going, how do I know that the smooth was going to be dominant? Well, experience, because I've known some of these questions before. But if you break down the question, you can figure this out too, even if you didn't have that experience, because it's heterozygous. Hetero means different. So if you're doing a capital S, let's do the parents. Heterozygous, we know is big S, little s, right? Heterozygous smooth pea. If this is heterozygous, we know it's big S, little s, and it tells us in the question it's smooth. All right? Now, if it had said heterozygous wrinkled pod, then that would have meant wrinkled is dominant. It doesn't. It says heterozygous smooth. So hetero means big S, little s, all right? And since it tells us it's smooth, then that means smooth has to be dominant. You have to think about this a little bit. So even though in this question it didn't straight up say smooth is dominant, wrinkled is recessive, you can figure that out by the question. Okay? Next part, wrinkled pea pod plant. Well, wrinkled is lowercase s. So that means it has to be homozygous, little s. Because if it wasn't homozygous, it wouldn't be wrinkled. It would be smooth. Okay? So capital S, lowercase s times two little s's. That's part A. That's our parents. And then the next question says do a Punnett square with it. All right, here we go. Break apart our parents. Big S, little s here. Two little s's across the top. Fill them in. Big S over, little s down. Big S over, little s down. Two little s's, very similar to our last question. What's the predicted genotypic ratio for the offspring? Genotypes are your alleles. It's the genes, okay? So what's the predicted ratio? It's going to be 50-50, right? 50%, you could do 2/4 or 1/2. 50% big S, little s. 50% little s, little s. Right there. That's our answer, genotypic ratio. What's the predicted phenotypic ratio? Phenotypes, what they're going to look like. What's the expression? Their physical traits, the physical appearance. So it's going to be 50% smooth. Sorry, my handwriting's so bad. I'm kind of writing vertical up here. 50% smooth. And then 50% wrinkled. 50% smooth, 50% wrinkled. Oof. Okay. There we are. All right. If this cross, letter E, I'm reading letter E. If this cross produced 50 seeds, how many seeds would you predict to have a wrinkled pod? Well, let's look at our ratios. We're saying 50% is wrinkled, okay? So half of the seeds would be wrinkled. So if I had a total of 50 seeds, how many seeds would you predict to have a wrinkled pod? What's half of 50? Say 50 divided by 2, and that's going to be what? About 25 seeds. 25 seeds. Or 50% of 50. Half of 50. How many seeds? 25 seeds. Half of that. Okay, number 2. Look at number 2. In cats, having hair is dominant to not having hair. Have y'all seen those hairless cats? I've had a friend that had one of those. And, it's different. If a male cat that's homozygous for hair is crossed with a hairless cat, what percentage of the offspring will have hair? So let's create our key over here. Having hair is dominant. I'm going to say a capital H would be having hair. And then we're going to say a lowercase h would be hairless. No hair. Okay. Now let's do our cross. If a male cat that's homozygous for hair, okay, homozygous for hair. Homo means the same, for hair. If they have hair, it's H. So I'm just gonna come over here to the side. Capital H, capital H. That's homozygous for hair. Is crossed with a hairless cat. That's gonna be lowercase h, lowercase h. What percentage of the offspring will have hair? Well, you probably can look at this and automatically go 100%. All of them. Because they're all going to get a capital H from one of the parents. But let's break it down so you can see it. So I'm going to put one parent down one side, so capital H, capital H. I'm going to put the other parent across the top, lowercase, lowercase h. And then I'm going to fill in my Punnett square. And then you can see that every single cat, every baby kitten that's produced from these parents is going to have hair. They all have the capital H, the dominant trait for hair. Now they're all carriers of hairless. So if one of these babies grows up and has a baby with a carrier, it's possible to pass that trait down. But for now, that trait is masked by the dominant hair trait, that capital H. So 100% is going to have hair. What percent of the offspring will have hair? 100%. Okay. All right. I'm going to move this down a little bit, or actually up for you. Sorry if I'm giving you motion sickness there, but I'm sliding this up so that I can work through the rest of these. There we go. It looks a little off. There we go. Maybe that's a little better. All right. Test cross. So we got another little topic here. Test. What is a test cross? All right. A test cross is when you don't know the genotype, the traits of a parent. So you might undergo a test cross and then examine the offspring. And then by looking at how the offspring turn out, you can make a prediction of what the parents are. The key thing to remember with a test cross is that you always cross the unknown parent with the recessives. Okay? You always cross the unknown with the recessive genotypes. And that way, by looking at the offspring, if that recessive is expressed, if we see a recessive pop in that generation, then we know that parent would be a carrier and be heterozygous. So let's look at this question. You found a wild black mouse. In mice, black fur is dominant and white fur is recessive. So let's do a little key over here. Black is dominant, so I'm going to use a capital B for black hair. And then it says white fur is recessive, so I'm going to use a lowercase b for white hair. Okay, one trait, one letter. So capital B, lowercase b. Letter A just says, using your notes, describe how to perform a test cross. This is defined for you in the notes and some of the earlier presentations that hopefully you've watched up at this point. But let's answer this together. So using your notes, describe how to perform a test cross. This is my own words here and my explanation. A test cross is when you're crossing an unknown parent with the recessive, a recessive parent, another recessive individual. Okay, so let's just kind of write this out. How to perform a test cross. So I'm just going to say cross the unknown parent with the recessive, cross the unknown parent with the recessive trait. I'm just going to use the word trait. Obviously, we're going to cross it with the recessive, in this case, a mouse. So we would cross the unknown black mouse with the recessive traded mouse. So what's the recessive trait in this particular question? Well, it's going to be a white mouse. So we're simply crossing the unknown with the white mouse. Okay, so let's look at this. Letter B, what are the two possible genotypes of the black mouse? Now remember, we don't know what the genotype of the black mouse is. We don't know. We just know it at least has the dominant trait. So what could the other allele be? That's what this question is asking. What are the 2 possible genotypes? We know it's got to have a capital B because it's black. What could the other allele be? Well, it could be homozygous. Sometimes we would call this like a purebred or a true bred form of that trait. So it could be homozygous, capital B, or it could be hiding that little b. It could be hiding that white trait. It still looks black on the surface, but it could be carrying the white trait because remember, the black color is dominant. So there's our 2 possible genotypes. So now letter C says, draw these 2 Punnett squares of a test cross, show both possible genotypes. All right, so we're going to cross big B, big B. Our first one would be big B, big B, right? The dominant homozygous with, remember it's a test cross, 2 little b's. Or we could be crossing big B, little b with our recessive traits. Okay, so we'll break these parents down, break these alleles down, big B down one side, 2 little b's across the top. Every one of these little mouse babies are going to be black. All the offspring is going to turn out to be black. Okay, let's do the second option. If the parent happens to be heterozygous and a carrier of the white trait, guess what's going to happen? About half of those babies are going to be what? White. Okay, so this is a test cross. We cross our unknown with the recessive, and then we examine the offspring. And if we see the recessive trait expressed in the offspring, then that means the parent has to be a carrier. The parent has to be heterozygous. If we don't see the recessive trait expressed, and every one of the offspring still expresses the dominant, then statistically speaking, the likelihood is that that parent is homozygous or a purebred, okay, a true bred of that particular trait. And in this case, it would be the black fur versus the white fur.